Abstract. Alkanes and alkanes are components of unsustainable fraction of the olive oil. These were analyzed by column analysis and are here proposed as an additional tool to certify the origin, authenticity, traceability, and chemical quality of olive oil produced in the Reggio Calabria province in South Italy. We have 9 cultivars were studied: Cananis, Coratina, Itrana, Licino, Nochiara, Utobratica, Pindolino, and Piculin, and lastly the Sinopolis. So they are grown in the region also in Calabria. Alkane in the range from 21 to 35 chain carbon atoms, and the alkenes in the range from 23 to 25 chain carbon atoms.
Oil of all cultivars showed a decreasing trend in total inalkane and inalkane content, with the oil of Sinopolis showing the highest content, varying from 328.50 to 314 milligrams and kilograms. Cultivars and harvest state significantly influence the inalkane and inalkene content. This finding can be useful to distinguish different olive cultivars and to decide the fruits harvest date for oil of the Reggio Calabria province or South Italy. A daily quantity of 30 grams of olive oil of the Sinopolis is the one with the highest inalkane and inalkene content. Was found to be in accordance with the suggestions of a European Agency for the Evaluations of Medicinal Product Comment for Veterinary Medicinal Products and Biogenetic Hydrocarbons intake for the human diet.
Olive oil is globally recognized for its biological properties due to its psychochemical composition. Calabria, South Italy, is the second largest Italian region for olive oil production, 755,032 tons in 2016. And for this reason, many works have been conducted to study olive oil produce. In this geographic region, with regard to the psychochemical properties of the fruit and oil, many of olive cultivars, and in particular with regards to the evolution during olive fruit ripening of dry glycerides and minor components such as sterols, fatty alcohols, and waxes. The unsaponifiable fraction also contains linear hydrocarbons among them, alkenes and alkenes, which have not been much studied, probably because of both European regulation and trade standards of the International Olive Council do not yet consider these components.
Alkane content was found to be affected by the geographic origin of the oil and can be used to detect enhanced concentration of leaf materials in olive oil because alkane profiles or olive leaves were found not to exhibit significant variation during the harvest season, in contrast to alkenes in olive oil.
Studies on vegetable oil reveal that alkane composition varies depending on the botanical origin, with alkane levels ranging between 21 and 34 mg or kilogram in avocado pulp oil in 19 and 32 mg or kilogram tomato in seals oil. To study hydrocarbon fraction, it is important to compare indigenous hydrocarbon with exogenous hydrocarbons. Alkanes is metabolized to fatty alcohol and acids into small intestine and liver, while mineral oil and paraffin migrate from paperboard to dry foods and from jute or sisal bags. Exogenous hydrocarbons are generally due to the non-voluntary addition, such as air pollution, incorrect storage, or incorrect processing and transport. The intake of biogenic hydrocarbon is typically between 10 and 100 mg per day.
Alkanes have also been used as markers in Alkanes have also been used as markers in nutritional studies with wild ruminant and non-ruminant animals. In recent work, they studied the influence of harvest season on alkane and alkanes content in virgin olive oil produced in the region of Calabria, South Italy, in a scientific context of scarce information with regard to the occurrence of alkane content in vegetable oils and in particular of olive oils. This is the first complete work studying the evolution of endogenous alkanes and alkenes during fruit ripening in a monocultivar olive oil and studying the effect of cultivar and harvest date on the alkane and alkene composition in order to characterize monocultivar olive oil and to have a further decisional element in relation to the date of olive harvest.